Thank you for watching our video. This is Dr. K's Genetics Fall 2018 class. My name is Stephanie.
I'm Anne.
I'm Vincent.
And I'm William.
So this experiment was an attempt to transform E. coli capable of changing isoamyl alcohol into isoamyl acetate by inducing it with a plasmid containing an ATF1 gene. This gene encodes a transferase protein that converts isoamyl alcohol into the isoamyl acetate, which produces a banana smell.
The reason why we want the banana smell is because
because uncovering these fundamentals of bioengineering can find applications and replacements for other industrial compounds such as paints, fertilizers, and perfumes. So let's get started with some of the introduction to it.
So Igem is basically used to facilitate our ability to cut plasmids at specific sites to transform it into a desired plasmid. Basically, we're going to call this plasmid A, or part A. The plasmid containing the promoter and RBS region was cut with ECO and SPE. The plasmid containing the ATF1 region was cut with XBAL and PST1. We then linearized the ampicillin-resistant backbone with ECO and PST to produce our desired plasmid containing the proper parts. After digestion, we ran each part through a gel electrophoresis to confirm that we indeed cut at the proper sites. After transformation of the E. coli, we placed it in an ampicillin plate to see if we got cell growth. As you can see, we got successful transformation by the colonies that were present.
So tube 1, since it's our transformed bacteria with IPTG and alcohol, is actually the tube we expect to have the banana smell. Tube 2 with transformed bacteria, no IPTG, which is the inducer, and alcohol, we expect no banana smell. And tube 3 with untransformed bacteria, IPTG, and alcohol, we also expect no banana smell. So after running a sniffing test with 7 subjects from our genetics class, 5 out of the 7 described a sweet or banana smell, and only 2 people described a bacteria smell in tube 1. In tube 2, which was transformed bacteria with no IPTG, everyone agreed that it smelled like bacteria. And in tube 3, with untransformed bacteria and IPTG plus alcohol, only 1 person said they smelled bananas. We believe this is because the mixture of alcohol, the isoamyl alcohol, and IPTG does create a sweeter smell. However, this smell will not linger because the bacteria is not transformed, and therefore the banana smell is not constantly being produced.
So while they were performing a sniff test with part of the transformed bacteria, we took the other part of the transformed bacteria and incubated it in a liquid culture. We then took this liquid culture and ran a diagnostic digestion. This means we cut at E and P to make sure that our plasmids came out to be the right size, and we found some pretty interesting results. For example, in this one, you can see that the uncut plasmid looks a little bit shorter than the cut plasmid, the digested plasmid. And that seems to be the case for all of the diagnostic digestions that we ran. This is because the circular plasmid supercoils and therefore travels faster throughout the gel than the linearized plasmids.
So one of the very interesting things that actually happened was that our plasmid is supposed to be around 4,000 base pairs, but when we only linearize the plasmid with 1 cut in either ECHO or PST, it turns out that the entire plasmid, or most of the plasmids, turned out to be around 2,000 base pairs. And because this bacteria with the plasmid is actually AMP resistant, we could infer or assume that it's somehow taken up a 2,000 base pair ampicillin backbone that somehow religated back together without taking any of the parts. Our results conclude that there are actually some bacteria that have taken up a fully constructed form of our plasmid. Due to the results of the gel, there doesn't seem to be a detectable amount of plasmid that were actually successfully taken in.
However, we could see that we had enough bacteria to produce the banana scent. We also noted that after 2 days of incubation, the banana scent was very minimal. And after 4 days of incubation, the banana smell was potent.
At this time, I think it's a good idea to address the questions that were asked during our presentation.
So, what is IPTG and how is this related to the LAC-I? Well, essentially, the LAC-I produces a repressor protein that prevents the transcription of the ATF1 gene. When introducing IPTG, which is very similar to allolactose, it actually binds to the repressor protein that's preventing the ATF1 transcription and promotes transcription.
The second main question we got during our presentation was, how did you perform the sniffing test? To perform the sniffing test, we got banana Laffy Taffy, put it in deionized water, shook it up, and got the banana smell. We then asked our subjects to compare the smell to our 3 tubes. Our first one being the transformed bacteria with IPTG and isomyl alcohol, which is the one that's supposed to smell like banana. Our second one was transformed alcohol with no IPTG and isomyl alcohol. And our third one was untransformed bacteria with IPTG and isomyl alcohol.
Produce the banana scent. We also noted that after 2 days of incubation, the banana scent was very minimal. And after 4 days of incubation, the banana smell was potent.
Between each tube and each trial of our sniffing test, we had our subjects smell some coffee beans in order to cleanse their palate. Although the majority of our results were consistent with what we expected, we did have one person who smelled bananas in the tube that was untransformed bacteria with IPTG and alcohol. And we did have a couple people who did not smell any bananas, who actually smelled bacteria in the tube that was supposed to smell like bananas. Further questions from the sniffing test, such as any allergies, any congestion, any sickness, could have helped us in determining why they smelled different stuff than what we expected.
So another question that was asked was, how did you deal with the satellite colonies? What we basically did was we found an isolated colony and we plated it on a different ampicillin plate to allow further growth. Another question we received was, why did we use ampicillin? Well, our part A and part B are in chloramphenicol backbone. We could have used other backbones like kanamycin and tetracycline, which we did, and our kanamycin backbone that we originally used did not grow any colonies. And so we changed to an ampicillin backbone that resulted in growth of the transformed bacteria. Some weaknesses that we found throughout our experiment was initially we did not have enough concentration of our parts. At this moment, we'd like to say a special thank you to Dr. Kirkpatrick, Professor Hines, and the entire biotech staff for helping us in every part of this experiment. We also want to take a moment to thank Igem for allowing us access to the parts that we desired. Thanks so much for watching our presentation. Yay!